It's my pleasure to introduce Professor Ross Hannan from the John Curtin School of Medical Research, where he's recently taken up a centenary chair in cancer research and as the new head of the Department of Cancer Biology and Therapeutics at the Curtin. His interests are in the nucleolus and ribosome biogenesis, particularly as they pertain to cancer, and his laboratory was the first to really prove that ribosome bioavailability was a rate-limiting step and a critical nexus that you can use to target cancer. And they're also the first in the world to specifically target RNA polymerase I and ribosome biogenesis therapeutically. So a very novel approach to targeting cancer. And he also has a sideline of interest in the ribosomopathies, as they're called, or defects in ribosome biogenesis that cause hematological disorders and predispose to cancers such as leukemia. He received his PhD from the University of Tasmania and then undertook postdoctoral studies in Philadelphia, returned to Australia in 2000 where he took up a position at the Baker Heart Research Institute and subsequently moved to the Peter Mac, where he was the head of the oncogenic signalling and growth control stream there, prior to last year taking up his position at the Curtin School of Medical Research. So we look forward to his seminar. Thanks.
Thank you. Thanks very much for the introduction and thank you for inviting me here to give a talk. I've got 2 pointers, different. One moves the slides, one points the thing. We're going to get into trouble at some stage, I'm sure. So I'm actually going to stand out here so I can actually see my slides. So we've had the introduction, we know where I'm working. Let's get the right one. So these are my disclosures before I talk. And so I thought I'd start at the end and come back to the beginning, and because this is sort of really directed towards postgraduate students, I thought I'd give them a bit of perspectives about how we went about developing this inhibitor, why we developed this inhibitor, and where we're going to now. So this is the end. This is the phase 1 trial that's undergoing at the moment at the Peter Mac for this drug, CX-5461, which targets a transcription factor or targets RNA polymerase I transcription. And it's driven by Simon Harrison, Grant MacArthur, myself, Amit Kott, and we're sort of 2/3 of the way through the dose escalation. So the question is, how did we get to that? The trials and the tribulations of how we got there. So I'll start at the beginning and we'll come back to the hallmarks of cancer, and this is the accelerated hallmarks of cancer. I guess this is the second generation. You're all familiar with these hallmarks of cancer, and indeed many people here in this institute have made major contributions to things such as resisting cell death, proliferative signalling, avoiding immunodestruction. I'm interested in transcription factors, and cancer is a disease of a genetic disease, and defects in transcription are a primordial regulator of this process. However, if you look at many of the therapies that are out there, and this is also taken from that paper, you can see things such as here, BH3 mimetics, driven through work done at this institute, cyclin-dependent kinase inhibitors. There's a paucity of transcription factors. And this is despite the fact that these transcription factors are directly mutated in cancer. Translocations in AML is a good example, or they're downstream of these signaling pathways. I guess this underrepresentation is disappointing, and I think it's really held back therapy in cancer, particularly because many of these transcription factors lie at the downstream events. There's a lot of problems with targeting signaling because there's redundancy in the pathways and resistance. And I think if we can target these, then we should be able to have a better approach to target cancer. So I guess the question is, why is that? I guess it comes down to the good, the bad, and the ugly. The greatest movie of all time. The good, things that you can target. We all know this: receptors, membrane proteins, enzymes, things which have small pockets, kinases, enzymes. Gain of function mutations. It's easier to inhibit a gain of function than to reactivate a loss of function. The bad, well, many things are bad. Intracellular proteins without enzymatic activity, loss of function mutations, P53, trying to reactivate P53. And the ugly, and this is the transcription factors. Most of these don't possess any enzymatic activity. Many of them are intrinsically unstructured, such as MEK, and they're really difficult to target. And I think this has been a problem. Now, it's a problem And if we just come back, that's not because there aren't interfaces to target, and this is just taken from one of our reviews. It's taking half an hour to work out which one of these to press. Pol 1, Pol 2 and Pol 3, you're all familiar with this. Most of us work with RNA polymerase II that transcribes the messenger RNA. Pol 3 is actually complex. There's 3 types of promoters there, and of course it transcribes non-coding RNAs. Pol 1, which makes the ribosomes. Now, if we just focused on Pol 2, there's a slew of different interfaces which you could theoretically target for cancer. If you look at the sort of the process of activation of transcription, and of course this is very simplified, it starts with transcription factors. Transcription factors dominate chromatin modifiers. In case people didn't think that, you reprogram stem cells with transcription factors. They can bind to their promoter elements. You can block that binding, protein-DNA interactions. Many of these transcription factors have domains which are masked. You could block that masking. They are post-translationally modified. You could block those post-translational modifications. These transcription factors recruit chromatin modifiers, which many of them have enzymatic activity, so we can modify histones. And so this is another area which you could target. Once you have these in the right conformation, you've remodeled the chromatin, you recruit the mediator complex, and of course, Pol 2 or Pol 3 don't have, Pol 1 and Pol 3 don't have mediator complexes because these complexes are thought to really what imbues Pol 2 with the ability to regulate complex processes such as differentiation. Nevertheless, this also is an interface which you could target with drugs, particularly because components of the mediator complex contain kinases such as CDK8. You form a pre-initiation complex, which is often paused until it's released from pausing, and these super elongation complexes once again provide a slew of different interfaces which you could theoretically target. Indeed, this is happening now with the bromodomain inhibitors, which are inhibiting the recruitment of some of these complexes to chromatin. Also, the CDK9 and the PTFP, which is also another target. So there's all these interfaces, but despite that, if you actually go and look at what drugs are in the clinic which target transcription, it's really limited, and there's almost no drugs which have been specifically developed to target transcription, probably with the exception of these sort of classic things like tamoxifen, all trans-retinoic acid, which, targeting these nuclear receptors, which are also transcription factors. And in fact, if you look at most of these drugs, most of them are falling into this general category of cytotoxic drugs, although it's clear that a lot of their action is by interfering with transcription factors through their ability to, say, cross-link DNA. So this is sort of really disappointing, and it's really, I think, held back therapy. Despite that, we've had a long-term interest in trying to target transcription factors, and particularly my laboratory and some of our collaborators are interested in targeting this hallmark of cancer, which is proliferative signaling, not so much the EGF receptors. And so we've sort of thought about how can we go about this, and my background was in transcription. I did my PhD thesis on MYC, and I've always been interested in targeting MYC. And of course, if you look at proliferation circuits in tumour cells, this is also taken from Weinberg, Hannan, you can see that MYC is one of the predominant drivers of proliferation. And indeed, MYC is deregulated in probably the majority of cancers, a bit like P53. Certainly amplifications are in probably 10% of cancers. And so a transcription factor that you'd want to target for malignancy is MYC. The problem with MYC, and there's many problems with MYC, we know it's dysregulated in the majority of human cancers and considered to be a major driver of malignant phenotype. And clearly, early work done here, Jerry Adams and Susan Corey have shown that MYC is sufficient to drive malignancy. And of course, it's become the holy grail of transcription factor-based cancer therapy. The problem, though, is MYC is intrinsically unstructured. It doesn't really have a structure until it binds MAX and other regulatory components. And so that's very difficult, particularly if you want to do structure-function-based studies. So the other alternative then is to go after, its targets. Of course, MYC hits 10% of the genome, so that in itself has been very difficult. And so what we thought very early on was that perhaps ribosome biogenesis would be a good surrogate. And if you go and do profiling and look at cells from earliest organisms, one of the major phenotypes or fingerprints of MYC is ribosome biogenesis. It's one of the primordial functions of MYC is to control ribosome biogenesis, and I'll come back to that. And so this suggests that perhaps targeting ribosome biogenesis might be a good surrogate to target MYC, and that's where all our studies really started, with this simple concept. So what about ribosome biogenesis then? And that's sort of really what we've studied for the last 10 years. So this is just a sort of an overview, and I just wanted to impart a couple of things about ribosome biogenesis, why we think it's a good target. So this is the process here. It requires all 3 RNA polymerases to make a ribosome, over 1,000 different genes involved in that process. And it starts with this process here, with the transcription of the ribosomal genes by RNA polymerase 1. This is all that RNA polymerase 1 does. It's the only known function for this enzyme. There's over 300 copies of these RNA repeats, found in head-to-tail fashion. These are transcribed by pol 1, they are then processed. They're assembled with the ribosomal proteins, which are transcribed by pol 2. They're assembled with a 5S, transcribed by pol 3. And then they're assembled as a large and small subunits. They're exported to the cytoplasm, where they're assembled into the functional ribosomes. So the point here is that this is 50 to 60% of all transcription in a mammalian cell. In tumour cells, actively highly proliferative tumour cells, it's estimated up to 80% of the total energy goes into this process here. So I guess the take-home point here is that you have to control this process. It has to be exquisitely regulated. And it turns out, not surprisingly, that the cells have developed many checkpoints to monitor this process. And I think that's what lies at the heart of targeting ribosome biogenesis. It's a furphy to think that we're going after proliferation. We thought we were, but probably the reason it has a therapeutic efficacy is because it's actually tricking these checkpoints, which I'll come back to. So MYC regulates all of these steps. It potently regulates pol 1 directly by binding to the promoter. It also transcribes the different components involved in ribosome biogenesis. So if you look at the pol 1 components, 90% of these are direct targets of MYC, and we've published that, compared to pol 2, which is about 10%. So there is a differential regulation of pol 1 compared to pol 2 and pol 3. So if ribosome biogenesis is downstream of MYC, then ribosome biogenesis should be involved in malignant transformation. And so what's the evidence of that? Well, the evidence goes back over 100 years. So well before people knew what the nucleolus was, it was the first subcellular organelle shown to be dynamic in terms of assembled and disassembled. But what was observed by these pathologists, that irregular and large nucleoli were an indicator of aggressive tumours. So this is silver staining, normal breast tissue. These dots here are the nucleoli where ribosome biogenesis takes place. This is normal breast tissue, this is breast carcinoma. You can clearly see how dysregulated they are. So that was one of the very first markers of aggressive malignant tumours. I think all tumours are malignant. So pol 1 transcription is also under control of these oncogenes, and I've already described MYC, but we know it's directly regulated by RAS, AKT, and other highly proliferative oncogenic pathways. So there's another reason to study it. What is less recognised is that many drugs in the clinic directly and indirectly target pol 1 transcription. So dactinomycin is a fancy name for actinomycin D. Low doses can selectively inhibit pol 1. 5-fluorouridine potently inhibits ribosome biogenesis well before it has defects in DNA. Many topo inhibitors are very selective for inhibiting pol 1 transcription. So I think we've always targeted pol 1 transcription, it's just we've done it with drugs which weren't selective. And of course, probably really importantly, the nucleolus, these nucleoli, although ribosome biogenesis takes place here, 60% of the proteins in here have nothing to do with ribosome biogenesis. They get sequestered here and released during the cell cycle. And many of these factors that are regulated by the nucleolus include p53. So if you mess with the nucleolus, you mess with many of these functions. I think that lies at the heart of targeting ribosome biogenesis for cancer therapy. So this, and I remember this clearly, I was recruited to the Peter Mac in 2003, interview with David Botel and Joe Trapani, and this is what I presented to them as my idea. So dysregulation of ribosome biogenesis contributes to the MYC-driven cancer phenotype. Targeting ribosomal gene transcription of the nucleus may serve as a novel therapeutic approach to treat a broad range of cancers. And I proposed in that meeting to demonstrate this feasibility, firstly through genetic approaches, and then through developing drugs. And it was an interesting interview. Dave looked at me with this sort of slightly nervous sort of, what is going on here? Joe Trapani was somewhat more supportive, but if you read his eyes, he said, at least the lunatic looks harmless. I mean, they were very sceptical about this process. I had a fellowship, I had funding at the stage, so I guess I was a fairly safe bet. But I think really what made the difference was that I was collaborating with Grant MacArthur and Rick Pearson, and Grant MacArthur had a long interest in studying MYC and Rick Pearson in studying the ribosome, and I think having these collaborations made a big difference. And for students out there, if you ever have to phone a friend, make sure it's a clinician scientist. It makes all the difference. And no, I'm being a bit flippant, but none of the work I've done would have would have happened without having a clinician scientist in that group that enabled all that translation of what we did and opened up many doors for me in terms of working with drug companies. This is the other reason they looked at me with this sort of, this is the pipeline for developing drugs. How are you going to achieve this? And we all know that there's drugs, assays, trials. We look at here the risk going down, but the cost going up. So on average, $1.2 billion to develop a drug. And of course, most of us work in this end here. It's 90% of investigator-driven stuff is here. And this is the so-called valley of death. How do you get your drugs from here to here, and how was I going to do that? And these are the things which had them significantly worried, had me worried as well. And these are the other things that had them worried. What happens if you inhibit POL1 transcription? Do you just get a slow-growing tumour cell? That's not so interesting. More importantly, can you get a therapeutic window, or are we just going to kill the patient at the same time as we kill the tumour cells? And remember, these were the days before we had things like proteasome inhibitors used in cancer. The idea of going back and targeting a housekeeping process that every cell requires was not really seen as being a viable mechanism, particularly that people had a lot of faith at that stage in modern approaches of high-throughput screening or even structure-function-based studies to go after selective mutations and the idea of personalised medicine. And so at the same time as I was looking for someone to help me with that, this drug company here, this is how it all started. Silent Pharmaceuticals was started in the States. And the two major focuses at the time of this company was targeting oncogene promoters and targeting kinases, PIM kinase and CK2. And so what's important here is their idea of targeting oncogenes is that it was known that these G quadruplexes, these 4G quartets, which form these particular structures, were found predominantly in many oncogene promoters like MYC, BCL2, VEGF, PGFA. And so if you could target those quadruplex structures by stabilising them, you might be able to selectively target these oncogenic factors. And indeed, they developed a drug called Floxin, which targets and stabilises G quadruplexes. And that actually got through to phase 2 trials, although I still don't know how because the half-life of this drug was 30 minutes. But I think it was a bit of positioning of the drug for marketing purposes. But what they found fairly soon that Quafloxin almost exclusively localised to the nucleolus and inhibited POL1. And it turns out, of course, that they weren't looking at the ribosomal repeats because they weren't part of the human genome sequencing, but probably the most enriched region besides telomeres for these G quadruplexes is the RDNA repeats. And they form different structures, and their drug happened to bind to the quadruplexes found in the RDNA repeats. So this drug fell by the wayside because of problems of getting exposure in patients and so on and so forth. But it sort of led them to the idea of perhaps targeting the nucleolus might be a surrogate for MYC. And they got in touch then with Grant MacArthur, who then passed them on to me, and that's how we really started developing POL1 inhibitors. And so we came up with the idea of trying to use small molecules to target RNA polymerase 1, predominantly then as a surrogate for MYC. We probably spent about $14 to $20 million on this, and a lot of this driven through Dennis Trojan. And so just to give you an idea of the company, these are the drugs that we're developing. This is the series of sort of funding that they received, Series D. So approximately $101 million was spent until we reached about 2013 where the company went bust and lost further funding. And a lot of that was because a lot of the money was pumped into these kinases because it was still very difficult to convince other financiers that targeting POL1 was a viable target. And I'll come back to that. The company was originally founded by Daniel Van Hoff, if any clinicians here. He's one of the probably most famous clinician scientists in the States driving clinical trials. And so what did we decide to do? So as I said, MYC is a potent oncogene because it drives all 3 RNA polymerases. And this is potent oncogenes and potent tumour suppressors are potent, particularly the transcription factors, because they drive all 3 polymerases. P53 inhibits all 3 polymerases. MYC drives all 3 polymerases. It drives POL2 transcription of the factors which regulate POL1 transcription. It drives 5S, and it directly binds to and regulates ribosomal RNA. So we decided to try and do screens to find molecules that would block this process. And so this is, I'm not going to go this in detail, this is the classic, this is what we wanted out of our target. We wanted an orally bioavailable drug. We're interested in oncology, solid tumours and blood tumours. We were after a selective inhibitor of POL1, and it was selective versus inhibition of POL2 and POL3, orally bioavailable, good half-life, and the other sort of characteristics, high therapeutic index that you'd expect. And so how did we do this? And I guess this sort of how do you go about screening for drugs? And of course, a lot of people in this institute are down at this end here where they're doing virtual screening and fragment-based screening because they know the structures of the drugs or the targets are after. Unfortunately, we didn't have any structures of our drugs. And so if you don't have structures, then you can't do this sort of screening. And what we decided to do was high throughput screening. And for that, we had a focused proprietary library, which had already been used for a number of other screens, had all the toxicals taken out of it. And we started the screening in 2006, and I'll tell you about that screening in a minute. And actually CX, or the progenitor of CX, not CX itself, was found in 2007 quite quickly. We screened other compounds after that, but CX, for a number of reasons, turned out to be the precursors of CX were the lead compounds. So this is actually how we went about this, and this is all done on a shoestring budget because we were trying to convince this company that this was actually a viable arm of their research. So the primary screen was simple: small molecule screen for inhibitors of POL1 transcription by qRT-PCR. And the counter screen was, well, it should inhibit POL1 but not inhibit POL2. And so we chose MYC as the counter screen, the POL2 transcribed gene, similar half-life. Really early on, we did PK analysis, and most often, because PK analysis is quite expensive, people do this much further down, but because we're doing this in a shoestring, we wanted to triage out drugs which were not going to have good pharmacokinetics early on. So we did this as a sort of an oral cassette where we actually took mice and put 5 or 6 different compounds into the one mouse, and then followed by a mass spec from blood, from bleeds, the breakdown of those molecules, how fast they're excreted, and so on and so forth. And this was really key to us early on identifying molecules that were going to be have a good PK. I think that was critical to the whole screen. We did a number of secondary screens once we had identified the small molecules and cell viability. And then we did the primary develop. We did a lot of cell-free assays to show that we're selectively inhibiting POL1 transcription, that we weren't inhibiting DNA synthesis, so on and so forth. And then we went through to in vivo screens where we did a number of different xenograft models. And here we had no idea what tumour to go after at that stage. So we picked off just standard xenograft models, which people do. Most of these were lung cancer and pancreatic cancer. Then we went on and did absorption, distribution, metabolism, excretion studies, and more PK studies, in this case in the mouse, microsomal studies to look at its processing by the liver. And then we did the in vivo screen, and these were full mouse studies and biomarker analysis. And back to more ADMET in this stage in rat and dogs, and this is probably the most expensive part of the screening procedure. This was done in the States quite clearly. And this was about $4 million to do the full ADMET and toxicology. And so that was done, and this is sort of the timeline, just to give you an idea of how that actually happened. So 2006 screening starts. Really, the bulk of the screening was completed in 2007, but it wasn't really until about 2010 that we were happy with this lead compound. And this was the first paper we published, so here's a sort of a warning. I had this period here where there was no publications came out from this work, which is problematic if you're running a lab because you've got to think carefully about who you put on that research. And clearly it's not something you can put students on, honest students on, or PhD students on because they may not be able to publish the data. And so most of this was done through really good research assistants and with me leading them and other people in the laboratory. So I had this first publication here. Didn't get any funding for this project until here. So all this was funded either through drug company or we borrowed and begged from other grants. So there's quite a long gap here, and I think that's probably not unusual. I guess the breakthrough for this project was really this publication here, which I'll tell you about, which really demonstrated you could therapeutically target POL1 transcription and get a therapeutic window. And then we got a development grant to conduct the clinical trial, and this is all hunky-dory, and we got the clinical trial developing here, but there's a lot of heartache, which I don't show here. So this is sort of the progression of the money. And really, the difference between here and here is really, a lot of this is all this toxicology studies. Xiling went bankrupt right here, right when we got the development grant. And this is because they were making other drugs, and the investment companies did not want to invest any money because they were really their lead compounds. I was jumping up and down the sidelines trying to drive development of our drug forwards, and it was just very difficult. So they went bankrupt right when we got this development grant, right when we were about to start the clinical trial. So we postponed the clinical trial, and we fought tooth and nail to try and get Senwa Xiling to sell CX-5461. And we were interested in buying it at the Peter Mac. I couldn't raise the money. The drug was eventually bought by Senwa Bio, a Taiwanese company which specialises in taking drugs, phase 1 trials, and then on-selling them. So the good thing is they rescued the project. The bad the downside is that they weren't interested in preclinical development. It's a bit short-sighted, so how do you stratify the drugs for patients? So basically, they bought this drug for $2 million. So this is what we spent on it, conservatively. They bought this drug for $2 million, and they got a free clinical trial paid for by NHMRC. Although we do have lots of, we had lots of IP, and particularly in the biomarker development. And so we eventually started the clinical trial in 2014, and that's ongoing, where there was a slight hiatus in the clinical trial because we had to reformulate the compound, but that's back up and running as of this month. So that's how we got to there. And as you can see, there was a lot of heartache in between. I'm going to skip that. So these are the original compound that we pulled out of the screen. We did a lot of SARS just to modify these different chemical groups, which we eventually ended up with CX5461, which is the lead compound. Clearly, it wasn't the most potent compound that we pulled out for POL1 inhibition, but it had all the right characteristics in terms of bioavailability, good half-life. So this was Floxin. This is CX5461. And the inhibition of POL1 IC50, 1.8 micromolar, we were down to 100 nanomolar. And you see all these different aspects here, selectivity versus DNA synthesis versus protein synthesis, much improved CX compared to Floxin. So we were encouraged because we got this to phase 2 trials. We think we should be able to get this further. This just shows you that this drug, if you put it on pancreatic cancer cells, you can inhibit POL1 transcription within minutes, as you'd expect for a small molecule, not affecting c-Myc as a POL2 gene. If you do RNA-seq and look to see how many POL2 genes are affected, this is 1 hour RNA-seq, increased gene expression, decreased gene expression. The red dots indicate variants induced in response to the drug. You can see there's very few red dots. So although this is 1 hour after treatment, we're totally inhibiting POL1 transcription, you have very little effect on POL2 transcription. Attests to the selectivity of the drug. As you move out in time, that's 30 minutes, 1 hour, sorry, you start getting responses, and a lot of these are p53 responses. We did a lot of studies, and I won't go into the details of working out where in the POL1 transcription does it affect. And by order of addition studies in vitro transcription, we could clearly work out that it blocks initiation. So this just shows you that here's our lead compound. Here's a compound almost identical with one difference here and here. This does not inhibit POL1 transcription at all. If you do shift assays to look for the ability of a labeled probe to be shifted when we add it to SL1, this is a core complex which recruits polymerase to the promoter. It binds to this piece of DNA, which is the RDNA promoter. So you can get a gel shift assay. If you try and use this drug, you get no shift. Test to the selectivity of the drug in inhibiting POL1 transcription. So then, just to summarise, I'll just go backwards. 3 different polymerases. We've targeted RNA polymerase 1, and we're doing chemical proteomics at the moment. It looks like the drug binds to TAF1A. It binds and blocks the ability of TAF1A to be recruited to the promoter. So it blocks a protein-DNA interaction. We do not have structure of TAF1A yet, and that's what we're going after at the moment. So what tumours to go after? This is a drug which targets generic processes. It's not intrinsically obvious what you'd go after. So we did this sort of curve here where we look at the dose-response curve. Shown on here is the IC50, cell death. This is a whole panel of drugs. You can't see these from me, but it doesn't matter. I'll just go through it in a minute. This is the median IC50, 147 nanomolar. You can clearly see is a massive difference in sensitivity, and this isn't just because some cells are not taking the drug up. Cells have a profound difference in how they respond to POL1 transcription, which we're still trying to understand. For the purposes of this, these are control cell lines here. These are immortalised but not transformed. These are all tumour cells. The most sensitive were the hematological malignancies, so that's what led us to go after heme malignancies. And this was really the first paper which looked at that. This was Megan Bywood, who was a PhD student at the time, and this was really her thesis work, where she demonstrated that if you inhibit POL1, you can selectively kill tumour cells by activation of p53. So we've already moved into another hallmark of cancer, which is tumour suppressors. And so the model she used for this, you'll know this because it was developed here. It's the spontaneous model of B-cell lymphoma driven by MYC, which replicates many of the aspects of Burkitt's lymphoma. And obviously the reason we chose that is because it's MYC-driven. I've just finished telling you that MYC is one of the primordial regulators of ribosome biogenesis. If you can't see efficacy in this model, you're never going to see it with this inhibitor. That was R. model. And of course, this is a good model system because you can GFP tag these cells and reconstitute B cell lymphoma by injecting the tumour cells back into the mice. So this is a typical sort of experiment. We just take these mice, we inject them with the B cells, reconstitute disease, and then give them a single dose of CX. This is an oral dose, oral gavage. And this is a maximum tolerated dose at this dosing regime once every day. We can show that within 30 minutes to an hour, we can potently inhibit POL1 transcription on the B cells from the lymph node. These B cells are GFP tagged, so the green cells are the tumour cells. And you can see quite clearly if you treat with vehicle, the tumour cells don't go away with time. But if you fax out the B cells from the CX, you can see that they disappear. This is one of those moments when you're in a career where you go, wow, look at that. There's a targeting ribosome biogenesis and there's a selective killing of tumour cells. This is within 24 hours, a single dose. You can significantly reduce the weight of these spleens. What was the mechanism? The mechanism was activation of p53 in this model system. We clearly rapidly activate p53 within 6 hours of treatment, even earlier, and you activate p53-mediated apoptosis. And importantly, it's not because they're running out of ribosomes. This occurs well before they have a ribosome shortage. It's a checkpoint we're activating, and I'll come back to that. So can you get increased survival of these mice? So this is a similar sort of experiment. Oral dosing once every 3 days, and you get a robust increase in survival in this model as a single agent shown here. So this is where the vehicle mice have to be sacrificed. This is the treatment window. This is CX. Unfortunately, and I'll come back to this if I have time, these mice do relapse, and this is a true relapse. If you re-challenge these mice, they no longer respond. So they are getting selectivity against this drug with time through clonal selection. But clearly, if you take mice early on in here, they're almost in complete remission. You almost normalise white blood cell count, and you can almost eradicate the tumours from the blood. But as I say, it does eventually come back. Somewhat disappointing. And a lot of our focus now is on combination therapies. It's p53 dependent. If you mutate p53, you get no response in this model system. So how do you kill these cells? What's the mechanism? It's actually nucleolar stress. And just to take you through nucleolar stress, if this is a normal cell, this is the cytoplasm, this is the nucleus, and this is the nucleolus where POL1 transcription is taking place and a symbol of the ribosomes, the ribosomal subunits before they're exported to the cytoplasm. And under normal conditions, p53 is kept at bay by the ubiquitin ligase MDM2. You're all very familiar with this. However, if you cause nucleolar stress, if you inhibit POL1 transcription, you stop the synthesis of this ribosomal RNA. So these ribosomal proteins are free to do other things. And it turns out that many of these ribosomal proteins, particularly L11 and L5, bind and sequester MDM2, which is no longer able to act on p53, and p53 accumulates. And p53 then activates either cell cycle arrest or apoptosis. And this is the fundamental basis of at least part of the therapeutic efficacy of this drug. Or so we thought. Sounds like horrible histories, doesn't it? So in any case, so this just shows you, if anybody's got kids and watch horrible histories, so this just shows you this in process. Here's this is a AML cell. This is a nucleus shown in blue, DAPI staining. These are the nucleoli. This is fibrillary staining, nice discrete nucleoli. You treat with CX, you basically disrupt the nucleoli. Fibrillary leaks out, fibrillary and other nuclear proteins leak out, including the ribosomal proteins, and they activate p53 within 1 hour. That's the mechanism of how we're killing those cells. And why don't you have effects on normal B cells? Because you failed to activate p53. So this is CX in a tumour spleen. We activate p53. If you take a spleen from a normal mouse, you fail to activate p53. And it's not because we can't activate p53, because if you gamma irradiate them, you can activate p53. So it's selective activation of p53 in tumour cells. So tumour cells are far more sensitive to perturbation of ribosome biogenesis than normal cells. And this isn't just a proliferative index. In fact, sensitivity to POL1 doesn't correlate with proliferation. So what about harder to treat tumours? And we've sort of been playing around with mixed lineage leukemia. So it's a form of AML. Median survival for these is 1 to 4 years. So these are, the outcomes for patients are very, very poor, and they are very refractory to standard therapies like cytarabine, doxorubicin. So we collaborated with Hans Zuber transgenic models. We used these, and of course these are driven by these translocations of these MIL fusion proteins. And so one of the prominent ones is the MIL-ENL translocation. So we have constructs driving MIL-ENL with also luciferase, and they also have activated RAS, because about 20% of these MIL leukemias have activated RAS. This just shows you can get a very good increased survival as a single agent in this MIL-ENL model using CX-5461, a maximal tolerated dose. Now, if you compare that to cytarabine doxorubicin, which is a standard therapy, it's far better as a single dose. But what was even more surprising, these are p53 null. I've just finished telling you it's all p53 dependent. These are p53 null, same tumour, p53 minus, and you still get a significant increase in survival. This was completely unexpected, and in retrospect, totally expectable, I guess, and I'll tell you why. So it turns out that if you don't have p53, the cells lock up in G2. So this is vehicle CX, they lock up in G2. What's even more surprising is the reason they lock up is we activate check 1 and check 2. So a lot of you will be familiar with the DNA damage pathways, single-stranded, double-stranded, activate ATM, ATR. Activate these checkpoints, evolutionarily it's thought, so that you can arrest the cell so it can repair the DNA damage. So we're activating these pathways. Drug company didn't want to hear about this because we're marketing this drug as a non-genotoxic drug. However, we've spent the last 3 years trying to demonstrate whether there is or isn't any DNA damage, and all the assays we've done, alkaline, comet assays, PCR across fragile regions, we see no DNA damage in the activation of these pathways. This is just a positive control CX. This is the positive control gamma radiation. These are the foci gamma H2X, the DNA damage. So, and I don't have time to go into it. Our data demonstrates that if you inhibit POL1 transcription, you activate this signaling pathway independent of DNA damage. We believe it's because you've got the most highly transcribed genes in the genome, completely devoid of polymerase, and this is a very unusual chromatin structure. And we've got a paper which we're trying to publish at the moment, which goes into the molecular mechanism of that activation. And so the other interesting point from these studies is that we were also able to get at the GMP population of cells, and these are thought to be sort of these leukemia-initiating cells, although this is somewhat controversial, but at least some of these leukemia-initiating cells, you might call them stem cells, are found in the GMP population, and we can clearly show we can significantly repress this GMP population of cells, both in the wild type and p53 null. And if you do these sort of serial transplantation experiments where you take a mouse and you treat it, mouse with a disease, you treat it, in this case it's AML, treat it, and then take those tumour cells out and transplant them serial dilutions into mice either treated with vehicle or CX. You can see that the mice which are treated with CX have significant increase in survival with the different doses, suggesting once again that you're getting at this leukemia-initiating population. And so that brings us to here, so this phase 1 study. So all that data, along with the, all the data on the PK and very good safety data led us to this clinical trial, and the primary objective is obviously to find the maximal tolerated dose, as it is with most phase 1 trials, but of course we're trying to establish whether we can actually see any evidence of biological activity. And so the patients in this are all patients with heme malignancies that have failed standard therapy, although the majority of the patients so far have been multiple myeloma patients, simply because they are healthier, sick patients. So we start off targeting this hallmark of cancer, malignancy, and in effect, our drug affects all these other, and I didn't really get into the DNA damage, all these other pathways. And I think this is why we get such good efficacy, it's because we are in fact targeting multiple pathways, multiple hallmarks of cancer. So I'll just summarise that, and I've got a couple more slides at the back end just to show you. So the response to POL1 inhibition is complex, and as I said, we should have realised this, it's one of the most complex processes, energy dependent, the cell has to undertake. It makes sense that the cell would evolve different checkpoints to monitor that process. Clearly there's a strong p53 checkpoint. If p53 is not activated, the cells run into a secondary checkpoint, and indeed if we take out all 3 checkpoints, we still arrest the cells in mitosis, suggesting there's further checkpoints we haven't identified. I didn't go into this, it promotes differentiation in those cell types, and I can talk about that later if anybody has questions. We think it targets the leukemia-initiating cells. Tumour cells are clearly more sensitive to these checkpoints. The million-dollar question is why? And so I think together all this is why we get such a good therapeutic response, although as I said, we do get relapse. So improving POL1 therapy, where are we going? I thought I'll just finish, I've got probably about 5 minutes. Yeah. So we're obviously doing rational combination therapies. The fact that we get resistance, acquired resistance, means this drug is probably never going to be used as a single agent. So what's a rational approach? What drug would you combine with a POL1 inhibitor? I think what promotes sensitivity and what promotes resistance, clearly clinicians want to know how you're going to stratify this drug, what patients you're going to give it to. And this is probably the most important question which we've been struggling with. And many of the ways to get around this is we've been combining our drug with genome-wide RNAi screens to try and identify the mechanisms of how we activate nuclear stress. How do we activate ATM-ATR? And I think they're best done through high-throughput screens. And that's really where we focused a lot of our studies. We've also done synthetic lethal screens to try and find out what drugs that have already been approved for use might work in combination with the CX and take a cell which may have a cytostatic response into a cell death response. And because I've sort of lost control of CX now, it's been bought out, and we've set up our own company to make second-generation POL1 inhibitors with some of the original people which we developed the first generation. And I had more control over this. The drugs are after smaller, they get across the blood-brain barrier, they have better PK. And we've got some lead compounds which are already in mice, and our goal is to try and get these into phase 1 trials by the end of 2016. A lot of the value of this clearly is dependent on whether CX makes it through phase 1 trials. And so the name of this company is Pymera. And I've got another company we're starting up at the moment, which is really targeting the nucleolus, and I think there's a lot of untapped area for research because the nucleolus controls so many cellular functions independent of POL1 transcription, and tumour cells seem to be very sensitive to perturbations in this process. So I thought I'd just give you a couple of examples. Rational combination therapies to delay response and extend survival. So as I told you, they relapse. Take these tumours as hemimic lymphoma, put them back into mice, they no longer respond. So a PhD student was actually the CDX conference recently described how TOPO2 inhibitors seem to confer resistance to this, but I won't go into that. But how do you get at those combination, how do you extend survival? So we've been combining it with inhibitors of the TOR pathway, and I'll go through the rationale. So POL1 transcription is here, it's downstream of growth factor pathways, including TOR pathways, AKT, PR3 kinase. So our drug inhibits POL1 transcription, prevents ribosome biogenesis. TOR inhibits signaling down this pathway and inhibits function of the ribosome. So the argument would be if you inhibit ribosome function and synthesis simultaneously, you'd get better effect in targeting these tumours. And we're also interested in this because TOR inhibitors and PR3 kinase inhibitors have been very underwhelming for solid tumours, at least in the clinic. We thought this might be a way to repurpose those drugs and add value. And this is just one example. These are B-cell lymphoma. Vehicle-treated mice come down here. Everolimus, the TOR inhibitor, has very modest effects. CX is better. Combine the two drugs and you get a significant increase in survival. So that's sort of a rational approach of why we've gone about targeting that process. And clearly we're applying this to solid tumours, and particularly prostate cancer, where MICs are currently overexpressed. Here's just another example. You've all heard of these bromodomain inhibitors, which are one of the sort of a few inhibitors out there really trying to target, I guess, elongation along with the CDK9 inhibitors. And that they sort of block these bromodomains binding to acetylated histone residues. And they're important because these appear to be important for these translocations that occur in these mixed lineage leukemias. And so, of course, you all know Mark Dawson, who published some of the really nice papers with these bromodomain inhibitors, I bet. And so we decided to combine these two inhibitors, which is sort of pretty neat because this is, at the most basic sense, you're combining a POL1 inhibitor with a POL2 inhibitor. You can't get any more further away from personalised medicine, I think, on the planet than that. The mice didn't melt, which was good. In fact, you get a really good increased synergy. So we've actually dropped the doses back of these drugs to look for synergy, right? So the doses of these drugs here are 15 versus normally they'd use 30, 25 versus 40. But you can see this synergy of the two drugs together. And in fact, you actually clear most of the disease from things like the liver and spleen. It relocates to these regions in the head, not in the brain, but in the head and the abdomen. These may be some immune privileged sites, but we think there is maybe some use in combining these POL1 and POL2 inhibitors. Very last two slides. The other way we're going about this is to try and do RNAi screens to understand these mechanisms of nuclear stress, because we want to find out what pathways may sensitise tumour cells. And we've done a whole bunch of screens, overexpression screens using the RVEC. This is with Tom Gonda, RNAi screens and Peter Mack compound screens. And I won't go into all these, I'm going to show you just two slides, siRNA screens. And so we did this screen. We asked, if you knock down a gene in the genome, does it activate p53? And is that mediated by nuclear stress? Because there's many ways of activating p53 in theory. Most of them aren't nuclear stress. So we did a high throughput screen, looked for genes that activate p53 and identified all these genes here, 820 candidates. All of these, this is the really remarkable point, all of these candidates require functional nucleolus. So these are all the hits. Well, these are all the genes we pulled out which activate p53 and require nucleolar stress. So if I inhibit nucleolar stress by knocking out one of the critical factors, they no longer activate p53. Our data suggests that all forms of activation of p53 require functional nucleolus, suggesting that avoiding nucleolar stress is probably a hallmark of cancer. So this is the primordial stages. What I have to go back is there's a mutation in MDM2 which blocks the interaction with ribosomal proteins, which blocks nucleolar stress. We're doing CRISPR-Cas to knock in that mutant as a secondary validation that all these factors which activate p53 when knocked down require functional nucleolus and are mediated to some extent through nucleolar stress, which I think attests to the importance of the nucleolus in terms of development, historical development, evolution, as a stress sensor. So I just thought of this, finish with this last slide about sort of, like I think, we screen for small molecules. The trouble with these small molecule libraries is they're really, really simple libraries. And they're not really going to be good for targeting transcription factors which bind to DNA interfaces. They have shallow interfaces. They don't have nice pockets. So I think, to be fair, it's been quite disappointing, the drug companies' ability to develop new therapies, particularly for pollen transcription factors, because they're based on this combinatorial synthesis. And the diversity of these drugs, including the compounds we use here and our compounds, are quite limited. And I think you really need to rethink this. And a lot of people do. And of course, this is what you're doing here with the HITS facility. And I think you and I have shown that the academic sector provides an alternative organisational culture for drug discovery, which can make real impacts in this area, at 1/10 the price of what drug companies are doing. However, I think if we stick going after these very simple, I'll start from here. It's okay. You can do virtual screening and fragment design if you know the structure of your drug. But if you're here and you don't know what the structure is, you're stuck with high throughput screening. I don't think it's going to work if we keep on using these simple synthetic libraries. At some stage, we're going to have to go back to the human chemome, natural products. Now, this has been a wasteland for drug companies. They've run away from it a mile. A lot of it was because they were going after target-based screens, where I really think the screens for these drugs need to be functional screens, where you go after a whole functional phenotype, like ribosome biogenesis. There's 1,000 genes that are targets there. You're going to end up with a target. The hard part is obviously identifying that target through chemical proteomics. I point out that 50% of the FDA-approved drugs have their origins in the global chemome. These are natural products. I think future have to somehow get a handle on natural product screens, and it's sort of where we're going at the moment with the Centre of Excellence we've just submitted. So I'll finish there. This is where I am. I'm at the John Curtin School of Medical Research. This is a brand new building. It's 4 years old now. I head the Department of Cancer Biology and Therapeutics. This is a new department, and our goal is to take fundamental research, translational research, and try and get this to the clinic. Currently, there's no phase 1 trials at the hospital in Canberra, so this is going to be a struggle, but we need to change that. So these are my 2 laboratories. I have an ongoing position at Peter Mac, and this is the people in my laboratory here. I've pointed out all the people that did the work as I went through on the bottom of the slides. This is my new laboratory at the John Curtin School of Medical Research. And with that, I'll take any questions. Thanks.
Thanks, John. Ross, really interesting overview of drug development from the ground up and how ugly transcription factors can make attractive therapeutic targets. Questions?
From the honour students first, right?
Yes, Amy? Yes.
Do you have any idea what the mechanism behind the increased sensitivity of tumour cells to the pollen inhibition?
Yeah, look, it's a really good question, and it's something we're struggling with because clinicians say, tell me which patients we're going to treat. So the most obvious one is tumours with elevated MEK. Lots of tumours have elevated MEK, and some of it's amplified, some of it's highly expressed. One hypothesis is that the reason MEK drives ribosome biogenesis is not just to drive more ribosomes, but it's to drive a strong, robust nucleoli, because that represses p53. In fact, we've got evidence now in that MEK model that if you inhibit POL1 transcription and block the apoptosis of expressing BCL2, those cells proliferate fine. So you can inhibit POL1 transcription by 50%. So it's nothing to do in those cells to driving proliferation, because ribosome biogenesis is in excess. It's about maintaining robust nucleoli and consuming all the ribosomal proteins into making ribosomes so they can't bind p53. So we think cells with elevated MEK is probably one of the sensitising mechanisms. The other sensitising mechanisms is the chromatin structure of the ribosomal repeats, and it's different in tumour cells to normal cells. A lot of these repeats get activated, and so we think a lot of it's to do with the chromatin structure, but that's at very early stages of development. But we think that's one of the major sensitisers. Yep.
Kind of a two-part question. I was wondering, I mean, obviously it seems you want to stratify your patients for an intact P53 response first. Are you doing that at the moment? And is it planned and also to target cancers where that's you're more likely to have P53? And then second, what happens to the normal mice that get the drug? Are they normal forever? Are there big side effects in them?
So, two really good questions. So your first question is about how do we stratify the patients for P53. So initially when we started the clinical trial, we thought it was P53 dependent, but of course I showed you that AML data where we were treating AML patients that were P53 mutant. So we don't think that is the case, but clearly P53 wild-type patients will respond better because they have 2 checkpoints we can activate. So we're not at this stage selecting patients based on P53, we're just trying to get to the maximum tolerated dose. When we get there, we'll back off and then we'll do an expansion cohort of patients where we'll try and select P53 mutant versus P53 wild-type. So we'll try and address how important that is. So I still think P53 patients will be more, P53 wild-type patients will be more sensitive, but it's not the only therapeutic mechanism. What was the other question? Oh, what happens to the mice? So we've done some models with VK-MYC-driven model of multiple myeloma and kept the mice on the drug for 300 days. And they died of relapse. They didn't die of toxicity to the drug. Now, we treated them once a week. Clearly, if you treat them at once a day, you'll eventually have problems. And one of the problems with high doses is gut compaction, probably because the highly proliferative cells of the gut, which are highly rapidly proliferating, are more sensitive to the drug. And you get myelosuppression. So the reason why the clinical trial is once every 2 weeks, once every 3 weeks, is that we do get suppression of the bone marrow, and so we wait for them to recover. So the idea is to go in and hit them hard with a single dose, back off, come back in 3 weeks later. So clearly, all cells will die eventually if you inhibit ribosome biogenesis. It's a matter of finding a good dosing regime with a high dose so you can kill the tumour cells and leave the normal cells relatively intact.
Given that you do see relapse, do you believe that maybe later stage tumours won't be as affected by this drug, but it only be the primary earlier stages that you would target?
No, we've only looked at the relapse in context of those emimetic lymphomas, which appear to be very dependent on P53, not those other checkpoints. And we've done deep sequencing, and it's TOPO2 mutations confer resistance to the inhibitor. We didn't understand that, and I was paranoid for a while, that's an understatement, that it was a TOPO2 inhibitor. And we've done all the studies, it's not, but then a paper came out in Science showing that TOPO2 is a part of the POL1 complex and required for POL1 transcription. So that sort of made sense as to why they may be resistant when you have mutations in TOPO2. I don't know what's going to happen in other patients which have very mixed backgrounds as opposed to these, model systems. So we don't really know what's going to happen to resistance. But clearly, like everything else, you select for clones that are resistant, we're going to have to do combination therapies. We thought going after ribosome biogenesis would be very difficult for cells to overcome that, but the power of clonal selection is enormous. John. You're not an honour student?
Yeah. Not quite. I might be devoted after this question. But going back more than 100 years, Paul Ehrlich was driven, magic bullet hypothesis, driven by chemical stains coming out of the German pharmaceutical industry. You mentioned that very strange structure for the nucleolar DNA, and particularly perhaps on that promoter region. Has anyone just done that very basic thing of looking for stains that would stain the nucleolar region? Because that suggests to me that they could also be something that would work.
No, most of them are those agrophilic stains, the silver stains that people have done. But that disorganised structure is because the chromatin of those repeats is different.
Yeah.
A lot of these repeats, there's 300 copies of repeats. In a normal cell, 50% are silent, and people don't understand why if they're derailing proliferation. And these tumour cells, most of them get opened up, although it doesn't contribute much to RDNA transcription because the density polymerase per gene goes down. There's a rheostatic mechanism. But the cells are addicted to this new chromatin structure, and we think it's for purposes which have nothing to do with POL1 transcription. So we've done a lot of 4C, looking at interactions across the genome, genome-wide interactions, and clearly a lot of genes interact with these open RDNA repeats. So we think there's something to do with three-dimensional structure of the nucleus and transcription that's important.
So in theory, it could be targeted by some sort of chemical compound that recognised that particular.
Absolutely, yeah.
And it would be very easy to screen for it. I mean, you're looking for a very specific pattern.
Yes. At the back, and then
Ross, you mentioned TAF1B as a target of CX. How good is the evidence that that is the target, and is there any other evidence for other targets of the drug?
Oh, I didn't say it was a target. Sorry if I may have not been clear. In combination, we've done combination therapies with CDK9, right? And CDK9 inhibitors, and they have profound effects in increasing survival. The caveat is that CDK9 probably regulates POL1 transcription directly. So I think CDK9 inhibitors are affecting POL2 transcription and affecting POL1 transcription. So the synergistic effect of targeting our drug, so our drug doesn't target CDK9, it's just when we combine it with a CDK9 inhibitor, we get increased therapy. We're not sure whether it's through POL2 or through POL1, but I think a lot of these chromatin modifying drugs and drugs that affect things like the super elongation complex, people forget that these factors are involved in POL1 as well. So at least part of their efficacy is probably through inhibiting POL1. Don't know if that answers your question, but.
What happens when you lose your drug and the biotech goes under and the drug gets bought by another company? Do you end up buying that drug off that Taiwanese company, or?
Oh, we tried to buy the drug when the company went bankrupt. We didn't know what was going to happen to CX, right? It's going to sit on the shelf. I tried. I couldn't get Peter Mac enthused enough to spend the $2 million. It's a lot of money, $2 million. So what happens if that drug had not been bought out? We would have had to synthesize the drug ourselves, and the cost of synthesis of the drug is extortionate for in vivo experiments. Plus, we wouldn't have been able to advance it to clinical trials because we wouldn't have had IP around it. So it would have brought all that to a halt. So we were lucky that it was bought out by Senwa, this Taiwanese drug company, although clearly their intent was just to get it to phase 2, and they've put not interested in preclinical studies. So that's the reason we started up another drug company, because I've sort of lost control of what experiments we do with the drug. Plus, anybody can synthesize the drug now because the structure's out there. So all that CK2, the checkpoint data, which we spent 5 years on, I just got scooped on 2 weeks ago because somebody synthesized the drug. They weren't under I was under restrictions of what I could publish with Senwa. Of course they weren't, because they just synthesized the drug. And they published all that data. Somewhat disappointing. So that's one of the things you've got to watch out for if you lose control of the IP around the drug.
Ross later on, actually.
You can ask now.
Yeah,
Okay, what I wanted to ask is, have you considered the resistance being due to the heterogeneity of tumours? So something that we saw really interesting during the symposium talk by Hans Peters was he had multiple subclones from a single cancer, and they responded differently to the same drug. And from there, he did some sequencing to see what genes were taking part in that resistance. Would something like that help identify which patients would be ideal to the treatment?
Yes, I mean, clearly, even in the emic lymphoma, which is meant to be clonal, I don't think it was clonal because we didn't pick up any p53 mutants to confer resistance. So I think they weren't new de novo mutations, or existing mutations already there, and there was some clonal heterogeneity. Yeah, probably. We're doing deep sequencing of all the patients. So if we are lucky enough to find a patient that responds and then relapse, seems to be very unlikely in a phase 1 trial, we'll sequence those to look to see if there's any clonal differences in those tumours to tumours that responded in the same patient. Although in a phase 1 trial that's pretty tricky, but yes, it's a good idea. It's something we'll follow up. It probably is clonal heterogeneity. Clones which prefer certain sites home to different sites, as opposed to an immune privileged site or something.
Come over time, but Ross is around for the rest of the day and having lunch with the students now, so just remains to thank you for a fascinating seminar.